Yes.
Okay.
Recording in progress.
Okay, let's get started. Yeah. So this afternoon I'm going to talk about cloning, but this cloning is very different than the animal cloning that you already know. But this cloning is to ensure that all cells in the culture are actually descended from a single pure cell line, which is all the cell in this culture are genetically identical. That means there is no mixed culture or contaminations from other culture. This is extremely important in tissue culture because you only want to culture a pure culture, right? So recloning established cell lines will frequently give a line with more consistent characteristic and result. Because if your culture are a resemble of many different types of cell lines, so you will not get the consistent characteristic because there are more than one cell line inside your culture. So you have to make sure that the one single culture is from the pure cell line, only one cell line in that particular culture. And cloning allows the screening of a large number of cell lineage and the selections of the cell line with desired characteristic. Okay. So it is actually very important. You can do a dilutions of the cloning, whereby when you have you culture the cell and the cell become confluent, for example, like this, right? And then you try to dilute them. You put trypsin here. You put trypsinize them because the cell are flattened at the moment, and you want to round it up, and that's why you put trypsin. And then you wash them and allow them to round it up. Then you do dilutions. The dilutions can be calculated, okay, by volume of the initial cell suspension on required, equivalent to the final cell concentration required over the initial concentration times the final volume required. Okay, this is the basic calculation for the dilution. And there are many disassociations procedures, such as you can shake off, you can do scrapping, trypsin alone, or you may do pre-wash with the PBSA, or you may use a mixture of the trypsin and collagenase, and then with this, or maybe you can use disperse, pronase, or DNS. These are all the procedure for you to do these associations. This association, because when the cell are cultured, okay, they become flattened and they are touching each other. And all of these association procedures is to help you to make the cell become individual cell. And later, they'll be rounded out, okay? So these are the procedure that you need to know, okay? For example, the fetus layer, this is not a particular, nothing really special about this because for example, when the cell, the rich confluence, what you need to do is to do subculture them, and then they become less cell, and then you can continue for the cell to grow. Okay, by providing more space and also fresh batch of nutrients for them to grow. All right, and in the semi-solid media, because some of the cell you have to culture in the solid media, for example, you extract the marrow from the long bonds and counts the nucleated cell, and you can dilute them, right? You culture them, and then when they become confluent, then you trypsinize them, and then follow the dilution, right? Follow the dilution, and then you can culture them for several weeks, up to depending on the characteristics of the cells again, right? Now, sometimes you probably need to do agar underlay. Yeah, agar underlay, for example, you want to do some dilutions. Yeah, for instance, if you want to do the agar underlay, if required, yeah, you need to get the diluted suspension cell with the agar, okay, the freshly prepared agar, and then you put it here. All right. So preparations of the agar underlays is very important. For instance, yeah, let me ask you, if you want to prepare one-time media, okay, with 0.6 agar from a stock solutions of 2-time media and 1.2% agar, how much of the solution I need to get from 2 times media and 1.2 agar? Any idea? Anyone want to try? No? Just keep quiet. No one really want to try.
Still thinking, doctor.
Thinking. You don't have to think, because when you see this, the answer is there. So easy. This is simple math. Let's see who get the correct answer. Okay. Ayo, ini sampai esok pun tak ada orang jawab ah. Are you really following me? Are you with me, or you just turn on your get your attendance, and after that you just leave it, then you are not in my class? Are you sure you are in my class?
Ya,
Yes, doctor.
Betul kah?
Yes.
betul.
Ha? Kalau komputer tak on, mana saya tahu kamu ada di depan saya ke tak? Okey, ada jawapan ke belum? Tak ada jawapan juga?
Is it we mix one part of the medium and the other part is the 1.2% agar?
Yeah, or how, for example, okay, that's correct. And how do you do it? You give me in the volume, for example, in ml, how much you get from the media to the stock media and stock agar?
Is it a 1-to-1 ratio?
Yeah, 1-to-1 ratio. That's correct. Okay, for instance, you get 100 ml of the 2-time media and 100 ml of the 1.2 agar, right? So at the end, you will get 1-time media with 0.6 agar. Siapa tidak tahu ini? Siapa tak tahu? Kenapa 1:1? Yeah, anyone? Semua tahu ah? Ayuh, hebat betul.
Tahu.
Ha? Tahu ah? Ya, kelas? So tak apa. So kalau you tahu tak apalah. Good. So it's 1-to-1 ratio. Okay. Since tidak ada orang nak jawab bila saya tanya, so I assume lah you sudah pandai lah. Ha? Okay. Dilutions of the agar for suspension cloning pun sama ya. Kalau kita nak prepare 1-time medium with the 0.3 agar for the final solution. So now, if we are providing you with 2-time media, okay, with 1.2 agar and ultra pure water, okay, so you should know by now. You probably need to figure out how much of the medium you need to take, how much of the agar you need to take, and how much of the pure water you need to take. So since everybody knows, they are so smart, so I don't have to explain lah. Because when I ask you, you don't respond to me, so I assume lah you faham, you pandai, right? So never mind. We continue. So diluting the cell in agar. So this is a serial that the dilute serially. Okay, in between 100,000 and 1,000 cell per ml. So this is 100,000 per ml. This one is 33,000 per ml. This is how you dilute it. You dilute it three times. Okay, so that you get 3 times less. Every time you do serial dilutions, you get 3 times less. Depend on the efficiency of the culture later, the plating efficiency later. How much of the cell that you want to plate? Okay, so you can plate it depending on the cell efficiency, right? Okay, and this is the suspension cloning in the metacell, and this is not just forget about this. You are not going to understand this. Okay, the other one is isolations of the clone with the cloning rings. Okay, it's very easy actually, because you just put a ring here, and the rings that's mean you only culture this particular cell. So the other cell you don't culture, so that you at the end you only get this specific cell, which are all genetically identical. So this is how you get the pure culture, right? So physical method of cell separations, there are many methods actually you can use, and successful separation techniques very much depend on the cell size, the cell density, the cell surface charges, the cell surface chemistry, the total light scattered per cell, and also fluorescence. All right, so these are the parameters that we are looking at for the successful separation techniques. Right, for this one, okay, the big size will definitely disseminated at the bottom, followed by the small speeds. Okay, because this is because of the gravity. Okay, so this is the cell separation by sedimentations of velocity at the unit gravity. The big cells will sedimented first, followed by the small particles, right? And this one is all cell separation by centrifugations, illustrations. This one is also very much depend on the density of the cell. Okay, and this is the one that you can see. This is the centripetal flow, and you can see the larger particle focus on the regions of faster flow rate, and followed by the small cell. Okay, and this is a centrifugal force. So that's why the big one will be sedimented here, and followed by the smaller cells. Okay, and these are also the same. This is the density centrifugations. As I said just now, the heavier one will sedimented first, and followed by the less heavy one, okay, or the light one. All right, this is the cell suppression of flow cytometry. All right, this is depending on the incidence of the laser beam here, okay, to separate the cell. Right, now it is very important to characterize our cell, and what are the characteristics that we're looking for. First is the species identification. We want to know which species, the lineage or tissue marker, the cell surface antigen, the intermediate filaments protein, the differentiated products, the enzyme, the special functions, regulations, lineage fidelity, and unique marker. These are the characteristics that how for you to characterize your cell. These are all the parameters that you need to look into for you to characterize the cells. I'm not going to talk in detail, because this will be your assignment 3 later. Okay. Then this is the example of the human or other animals cell. Okay. From under the microscope, I'm not going to talk about that also, but this is a cell line selection. How you select your cell line. Okay, you must know how to characterize your cell, and you must know how to select your cell line. That's very important. So when you want to select your cell line, you have to look at the function requirement. What are the functions that is required for the experiments? Whether the cell is from the finite cell line or continuous cell lines, normal or transformed cell line, which species the cells is originate from, the growth characteristic, will it be fast growing or slow growing, availability, is the cell easily available, validation, has the cell been validated or not, do we have any markers to validate the cells, phenotypic expressions, the control. Do you have the control? If you want to, how to say, to do the experiment, is there any proper control, and how stable the cell is? Okay, so these are the cell line. So there are two parts in the assignment 3 later, which is how to select a cell line and how to characterize your cell. Okay, so I'm not going to talk that in detail because this will be your assignment. Read more. Okay, read more because this is particularly very important for you to understand. Okay, as a young scientist, you should know how to select your cell line and how to characterize your cell line. And freezing and thawing is very simple. All right, so right, this is the isolation. Okay, this is the isolations of the organs. Okay, how can you establish a pure cell line? So when you isolate an organ from certain animals, okay, and you wanted to get your cell lines of desired, so what you do is you dissect them. Okay, you get the organs, you dissect out the organs, excise the organ, and then selections of the tissue required by removing the fatty acid and other necrotic tissue. Necrotic tissue is other dead tissue which you don't want in your cell culture. And after you do the dissections, you can do 3 different methods to generate your cell line. First is the fine dissection. Fine dissection is how you chop the cell line and to get your explant. And after that, you establish your explant, and then you outgrow them, and you transfer the explant, or you subculture them and outgrow them, and at the end, you get your cell line. Second is by mechanical disaggregation. All right, so you may use sieving, syringing, or vigorous pipetting. Okay, this is particularly good for the soft tissue like our liver. So you can disperse the primary cell line. Then after that, you do subculturing for several times to get your pure cell line. Or the third method is the enzyme disaggregation. You may use colchicine, warm trypsin, and also collagenase. And colchicine, you need to incubate them overnight and then short incubation time. But for warm trypsin, you probably need long incubation time, but with sampling. For collagenase, you may need long incubation time in the complete media. For warm trypsin and collagenase, you need to centrifugate them. But for colchicine, you don't have to. Then after centrifugation, then you resuspend them and reseed them. After that, you reseed them, you establish your primary culture. After the primary culture, then you continue to do your subculture for several times until you establish your cell line. And this is how we get the cell line from an isolated organ. All right, are you with me?
Yes, doctor.
Yes, doctor.
Okay.
Yes, doctor.
So very important that there are 3 methods here: fine dissection, mechanical disassociation, and enzyme disassociations or disaggregations for you to establish or to cell line, or how you generate the cell line from the organ that you isolated. Okay. And another one is right. Another one is the disaggregations of the primary of the culture and the primary culture. So primary culture, as you all know, is a culture started from a cell and tissue or organ taken directly from an organism or before the first subculture. And the explant is a fragment of the tissue transplanted from its original site and then maintained as an artificial media. So if we want to establish a mouse embryo culture, so what we need to get is you kill the mouse by vertical dislocation and swab. Like this one, I don't have to read lah. Let's look at this. Okay, first you swab the abdomen. Okay, with the tissue containing a 70 or 75% ethanol. You swab them, right? Then after that, you tear off the skin. Okay, right in the middle here, you tear off the skin. This is not right. Huh? You should wear glove, but this one they did not wear glove. You should wear glove. Then you tear the skin to expose the abdominal wall. This is the abdominal wall. Okay. Then right after that, then you use a scissor and another forceps to hold it, and then you open the abdomen. Right, abdomen, and to expose the uterus in situ. The bits here, this is the small light structure is what the embryo content. Okay, and this is the embryo. You isolate the embryo, remove the uterus, right? Then you place it on the embryos. The embryos on the petri dishes, and you remove the necrotic tissue or fatty tissue and other membrane which you don't want. Then after that, after removing the membranes, you know, the necrotic tissue, the fatty tissue, and other unwanted tissue, then you chop them very finely, and then you culture them. That's how you establish the mouse embryo cell line. All right, are you with me?
Yes.
Yes,
Doctor.
Okay, and this one is you establish the embryo, the cell line from the egg embryos. Okay, from the embryos, same thing. So what you need to do is you swab the egg with the 75 or 70% ethanol. After that, after swabbing, it's very important to swab it because you don't want this is how you do the aseptic methods. Okay, then after that you crack the top of the cell and peel off the edge. Usually you crack off the blunt end because egg they have blunt end and also the sharp end, isn't it?
Yes.
Yeah, so the blunt end actually contain the egg sac, so you crack at that side, and then you try to peel off the top shell. Then after you peel off the top shell, then you can see that to reveal the colloidal membrane. This is a colloidal membrane, and after that you remove this one, you pierce the colloidal membrane and remove the membrane and get the embryo out, and then you do the same thing like the mouse embryo by removing the unwanted tissue, the necrotic tissue, and fatty tissue. Then after that, you chop in very finely and proceed with your culture. Right?
Recording stopped.
All right. So that's all for today's lecture. Any question, class? It's very short actually. So by finishing this lecture, that means we are done with the first 7 weeks, and by next week, Dr. Hall will take over the class, and this Saturday will be your midterm exam, right? Yes, Geraldine, do you have any question?
Yes, doctor.
Yeah.
Regarding the dilution question just now, I don't really get it.
Why don't you ask just now when I asked you?
Sorry, doctor. I was quite blur.
Yeah, you students are very blur, you know. When I ask question, you just keep quiet and keep quiet and keep quiet. Then when I almost finish the class, or when after the class, then you busy text me. I don't know why students is like that, you know. You should ask when I ask you the question, okay? Or you should ask me if you don't understand when I talk about that. Which one? This one? Or
Yes,
which?
doctor.
This one? Or this one?
Yeah. Just now that one.
This one?
Yeah.
Okay. It's very simple method. Do you know M1V1 equal to M2V2?
Yes, doctor.
Okay. It's very simple. Okay. If you look at this is 2-time media and this is 1.2 agar. So the final solution here is 1-time agar with 0.6 agar. Okay, how we establish this is very simple. You get one to one by looking at this diagram. Actually, is 1 to 1 is very simple because, for example, I take 100 ml from the 2-time media and 100 ml of the agar media. So the end solution here is 200 ml. Betul tak?
Yes.
How much of the media inside here? 100, right?
Yeah.
100 over 200 equal to 1 over 2, right?
Yeah.
1 over 2 times 2.
1.
1 time kan?
Oh.
How much of the agar inside this final solution?
Is it 1?
100 ml in 200, right? Because the final
Yeah.
solution is 200, right?
Yeah.
So 1/2 times 1.2.
0.6.
Ya, 0.6. So apa susah dia? Right? So it's one part of the media and one part of the agar. Correct?
Yeah.
So if student keep quiet, I assume you know. Okay? Although I know you don't know. But because you don't want to admit you don't know. So I'll go very fast. Okay? This one also the same. Yeah? For example, to prepare 1 times of the media with 0.3 agar. How do you prepare that? It's very simple. Yeah, you take two parts of the media, 1 part of the agar and 1 part of the ultra pure water. So for example, I take 200 ml of the media, 100 ml of the agar and 100 ml of the pure water, ultra pure water. So my end solution here is 400 ml, isn't it?
Yes.
So 400 ml, I take 200 ml. So 200 over 400 equal to 1/2, right?
Yeah.
Yes, doctor.
Yeah, 1/2 times 2x. 1x kan? Betul tak?
Yeah.
Ya? Tidak, Bang. Saya rasa susah nak ajar kamu orang ni yang tidak mengaku. Sekejap. One moment. Right, can you see this?
Yes.
Yes, doctor.
Okay?
Yes.
So I take 200 ml. $100 ml. 100 ml. So the final solution is 400 ml, right?
Yes, doctor.
In here.
Yes.
So how much of the medium I took? 200, right? Betul tak? Untuk yang medium ni.
Betul.
Okay? So for agar is 100 over 400. Betul tak?
Yep, correct, doctor.
Betul.
So this is simple mathematics actually. Mana ada susah. You also can use M1V1. Boleh juga, tak ada masalah. You just plug in the number and you get it already. Pernah tengok M1V1 equal to M2V2?
Yes, doctor.
Pernah.
Pernah kan? Senang saja sebenarnya, mana ada susah. Right? Cuma you kena pusing sikit otak tu. Tak ada yang susah. But I have to admit lah, biasanya pelajar yang ambil biotek ni dia punya matematik memang lemah. I have to admit that. Kalau you matematik sudah, kalau you are good in maths, you are not taking biotech, you definitely will go to engineering or you take pure maths lah. But we have to admit our weaknesses lah. But a lot of time because of the ego, we don't admit our weaknesses. But anyway, so siapa ada internet issue kalau kita buat exam pada waktu malam? Ada tak internet issue?
No, sir.
Yeah,
I have internet issue if it's raining.
if it is raining lah.
Yeah, right, like now.
Never mind. It's okay. We will understand that. We understand that. You just finish. If you need more time, you let me know.
All right, doctor.
Okay. If you need more time for, if your internet is not stable and then your exam progress is affected, you let me know. Maybe I can extend another 15 or 30 minutes. Let me know. Okay. Jangan biarkan tak bagi tahu saya apa-apa. Okay. Just text me. Or text in the group. Okay.
Doctor,
Okay, doctor.
Yeah. Ada
if
soalan?
the internet connection while we're doing the exam is like it breaks, will we still be able to continue the exam
Yes.
in the same page?
Yes. Boleh. Dia akan save your answer.
Okay, thank you, doctor.
And the exam, once you buat, you tidak boleh pergi balik, you cannot go back. Maksudnya, kalau you buat soalan 1, 2, 3, soalan 4 you tak pasti. So you skip soalan 5. You tak boleh balik ke soalan 4 dah.
Oh, okay, doctor.
Okay? You please make sure everybody know this, because this is to avoid anyone to discuss the question. And all the question that everybody see is different. For example, what Zacchaeus see with Lindbergh see is different. And the answer also different. So ini untuk mengurangkan, meminimalkan pelajar meniru lah ataupun discuss semasa exam. We have to do this, okay? Because biasanya pelajar akan discuss lah, walaupun kita tak boleh discuss. But you know lah, siapa tahu, okay? Hanya Tuhan akan tahu mana satu pelajar yang jujur. Yeah, I don't want to penalize anyone, but you should be honest to yourself. Okay? Because the exam is to test your understanding and not my understanding. Okay? Boleh tak?
Okay, doctor.
Boleh,
Boleh,
doctor.
Okay. So kalau macam tu, then habislah kita punya tissue culture untuk semester ini, untuk first 7 week. Then minggu depan pukul 10 pukul 2 petang hari Selasa, then it will be Dr. Ho. Okay? So all the best for your midterm. Okay, siapa nak ada orang
Doctor.
ada orang siapa nak ada orang nak attendance kat sejak. Siapa nak menu attendance tadi?
Doctor, I need manual attendance.
Siapa?
Natalia.
Oh, Natalia. Okay.
Thank you.
Siapa lagi?
Nura Fiza.
Nura Fiza, and then Yo kah?
Yeah.
Yo, okay, done.
Okay, thank you, sir.
Okay, so all the best to you all, and take care and stay healthy. All right?
Okay, doctor.
Take care.
Okay, take care. Bye bye.
Thank you, doctor.
Bye.